Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth and that's biology. Yes, hello Vishal, a very happy good evening. Yes, Vairavan, hello. Yes, I'm good, thank you. How are you? So thank you so much for joining my lecture. So please do like, share and subscribe. Yes, good evening. So today we are going to talk about the leftover bits of cell, the unit of life, as well as some important questions. So only when I was making the PPT, I realized, yes, there are some more points that I didn't tell you. So we will first complete the leftover points and then we'll move to question. So I didn't get 30 questions, but I got much lesser questions. So let's see if you can answer them. Okay, but with this we will end. So today will be the last lecture of this chapter. So after this I will move to biomolecules and then quickly move to your had cake, no Vishal. So then I will move to cell cycle and cell division. Okay, no, I didn't have cake, so where will I get cake suddenly? Huh? Keshav is not baking cake for me, so I will not get cake suddenly. Okay, all right. So before we go into today's lecture, so announcement for tomorrow's lecture. So here we go. Yes, so this is my schedule tomorrow. So we have small portions of photoperiodism, vernalization here. So we will finish this and we will discuss important questions from your plant growth and development as well. Okay. Yes, so next chapter in line will be biomolecules. So I will be starting biomolecules from Wednesday. Okay, Vairavan? Yes. And tomorrow we are having a mentee, so it is just the chapter, it is episode 7. So it can be any chapter or a combination of chapters, so please prepare for the same. All right, yes? Okay, so this is my schedule for tomorrow. So without any further delay, so let's start talking about cell, the unit of life. So this will be the last lecture, so I will not move forward. Guess the chapter. Yes, so it's going on. So I guess you didn't join in between. Is that so? Yes, Satyendra. Hello, hi. A very happy good evening. Thank you for joining my lecture. So please do like, share and subscribe. So these are the points that we were talking about. So if you remember, so last class we were talking about your basal bodies, your centrioles, etc. Yes. So we were talking about your ribosomes. So after that, we went on to talk about your cilia and flagella. So the point where your cilia and flagella arises, so that is known as your basal body. So that has centriole-like structures, right? So how are they? Like the cylindrical shaped structure, and this core is known as axoneme. So this has 9+2 arrangement. What is this 9 and what is this 2? So they are the microfilaments or microfibrils. So on the periphery, you have 9 microfibrils. So like this. Okay, so in the center you have 2 microfibrils like this. So that is why it is known as 9+2 arrangement, and this is known as your central sheath. Fine. So they are attached to the base. The periphery is attached to the all are missing. Yes, so the peripheral microtubule is attached to the base via what is known as your radial spokes. Okay, so these two are interconnected via your bridges. Okay, so this is what we've been studying. So this is your 9+2 arrangement. They will be at sharp 6. Okay, so it happens that they miss out. Let's see if they come. So then we have your centrosomes. So centrosomes are present only in case of your animals. So they contain centriole-like structures. So your centrioles are cylindrical structures, but they lie perpendicular to each other. They lie at 90 degrees to each other. Okay, so that is your centrosomes or your centrioles. So how is this core arranged? So they also have 9 microfibrils, but in the center there is nothing. So it is 9+0 arrangement for your centrioles or your centrosome. Okay, yes. So this 9, so you have 9 like this. So each one has a triplet or 3 bundles of your microfibers. So each of the 9 is a triplet or it has 3 microfibrils. Okay. So that is 9 plus 0 arrangement, and the center portion here is known as a hub. Okay. Is this clear? Fine. So this is your 9 plus 0 arrangement. So then we went on to talk about your nucleus. Yes. So if this is your nucleus, so on the outer side it is attached to the endoplasmic reticulum. So this is attached to ribosomes. So this is your rough endoplasmic reticulum. So these are your nuclear pores. Yes, hello, good evening. So thank you for joining my session. So please do like, share and subscribe. So this is your nuclear envelope. Okay. So this is your nuclear pore. So nuclear pore is controlled by annulus. That is a complex of nuclear pore proteins. Yes. So inside this, the space inside your nucleus is known as your nucleoplasm. So in your nucleoplasm, you have your chromatin material scattered like this. So in your chromatin material, so apart from this, you have what is known as nucleolus. So nucleolus is the ribosome factory. So nucleolus produces your ribosomes. So this is the structure of nucleus. Is this clear? Yes. So come on guys, please do like, share and subscribe. Please like the session. So this is the structure of nucleus. So this is where we stopped. Okay. Yes. So today let's talk about what are or some parts of your chromosomes. So what are chromosomes? What are chromosomes? So our genetic material is DNA, right? So DNA is a polymer of nucleotides. So you have A, T, G, C, so long and so forth. So this DNA is 2.2 meters long and it has to be compacted in a cell which is 10 power minus 6 meter diameter. So it has to be packaged. So how is your DNA being packaged? So before that, any portion on your DNA, okay, that codes for a sensible product. So this is known as a gene. Right? Yes, Shimpi. Hello. Yes, a very, very happy good evening. So yes, I'm good. Thank you. How are you? So thank you for joining my session, Shimpi. So please do like, share and subscribe. Fine. So now how is the DNA packed? So I hope all of you know your DNA is negatively charged and it is acidic. Your DNA is negatively charged because of the phosphate group that is present. So DNA has a 5' phosphate and a 3' OH group. So because of this phosphate, it is negatively charged and it is acidic as well. So your DNA binds with a group of positively charged proteins known as histones. Okay, so how many histones we have? So we have 5 histones, right? Hydrophilic molecule, yes, you're right. So as you like, I will explain on blackboard. So you have histone proteins. So how many histones we have? H1, H2A, H2B, H3, H4. Okay. So this H2A, so 2 molecules of H2A, I write like this. Then you have H2B, you have 2 molecules of H2B. Then we have H3, so 2 molecules of this and 2 molecules of H4. So totally it forms 8 molecules. So this is known as your histone octamer. So this octamer repeats itself. So if you have repeating histone octamers on top of which your DNA is wound like this. Okay, so this is the first level of your histone DNA packaging. So this is your DNA. Yes, and this is your histone octamer. So here I have my H1 histone. So again here I have my H1 histone. So this structure of DNA wrapped around histone proteins is known as your nucleosome. So repeating structure of nucleosome is called as chromatin. So chromatin is further organized into solenoid structure, that is beads on a string. So solenoids further form loops. So loops further form your chromosome. Clear? Yes, surrounded by H1. So H1 is the linker histone. So this structure is called as nucleosome. Yes, Dharani, a very happy good evening. Yes, Anthony, happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So is this clear? Yes, this is clear for everyone? No doubt? Okay, so that's okay, Antonia B, that's fine. So this is how your DNA is packed. But why am I saying this? Because now we are going to talk about the structure of your chromosome. So how is your chromosome structure? So what is the difference between this and this? What is this and what is this structure? Yes? What is this structure? What is this structure? Both are chromosomes. So whenever you have only one center point, so this center point is known as centromere. So whenever there is only one centromere, irrespective of how many ever arms are attached, so this is also a chromosome, this is also a chromosome. So these are known as chromatids or arms of chromosome. So there's only one centromere. So this is also a chromosome. This is also a chromosome. So here you have the genes, because inside the chromosome, what you have? Nothing but your DNA, right? So on the chromosome, you have genes, right? So this is your centromere. So centromere is where the chromatids are attached together, okay? So in the centromere, you have disc-shaped structures like this. Okay, in the centromere, you have disc-shaped structures like this. So these disc-shaped structures are called as kinetochores. So kinetochores are where or is also known as the point of attachment of your spindle fibers. Okay, yes. So this is the structure of your chromosome. Hi Keshav, you're late. Okay, so I guess we decided that you slept off after having cake. Is that so? Yes ma'am, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Yes, kinetochores, that is where your spindle fibers attach. So this is the structure of your chromosome. So why am I teaching this? So this is also part of this chapter. So I don't want to leave any portion uncovered. So I am covering it, after which we will go into questions. So this is clear for everyone? No doubt. What is this structure? Everything is clear. So whatever the terms that I have written on the board, is it clear? Yes, any more doubts? Can I move forward? Yes. Can we move forward? Can I go back to my slide? Yes, everyone, it's clear? No doubts? Okay, so moving forward. So this is how your DNA is packed. So this is what I was saying. Okay, all right. So congratulations and all the best to your brother. Okay, Kesha? Yes, so this is how your chromosomes are packed. So moving further, so histones are positively charged proteins. So it is made up of lysine and arginine. So they are basic amino acids. So they are basic amino acids, all right? So you have so many types of histones. So this is your linker histones and others form histone octamer. So like this. So this structure is known as a nucleosome. Done? Okay, so this is why I wanted to talk about chromosome. So in your body, you have a very unique mechanism. Your cell is very clever enough, which I have already told you n number of times. Apart from that, there's something else which your cell does. Your cell, what it does, it has regulated gene expression. So what do you mean by regulated gene expression? So this topic comes in your molecular basis of inheritance, under which we talk about your lac operon, if you remember. So what does this mean? If your DNA is coding for 35 or 25,000 genes, so 25,000 genes, if it codes, you have 25,000 products. But at the time, you will not need a lot of them. Yes? Serotonin and histamine allergy, yes, okay. But at the time, maybe you need only 500 gene products. So your cell will not spend time in translating or transcribing these many products, okay. So you have something called as central dogma of life. So what is central dogma of life? So you have your DNA. So your DNA replicates during the S phase. Then the information on the DNA is copied onto the mRNA. So this is transcription. And then finally mRNA is translated into protein, right? So this is translation. So if at a time only 500 gene products are needed, it will only transcribe or translate these 500 genes. It will not translate all the 25,000 genes. So how is this made possible? So the genes which are to be transcribed and translated, they are loosely arranged. So the genes which have to undergo transcription and translation, they are loosely arranged and they are known as euchromatin. So every time the DNA's information is copied to the mRNA or the DNA is being replicated, so DNA is wound around histone. So while replication and your transcription, your DNA unwinds from the histone octamer, it undergoes replication, transcription and once again winds back on your histone octamer. So that is how this process happens. So central dogma was given by Francis Crick. Okay, Anthony? Okay, so such a chromosome is known as euchromatin, which is transcriptionally active, meaning it is going to give a product. Suppose the cell does not need the particular product, so those will be tightly arranged around your histone octamer and they are known as heterochromatin. So they are transcriptionally inactive. Okay, so did you guys understand what is euchromatin, what is heterochromatin? Yes? Did you guys understand what is euchromatin, what is heterochromatin? Should I explain once again? Or is it clear? It's a part of your chapter. I am not teaching anything extra. Yes, clear the term euchromatin and heterochromatin? Everyone is able to comprehend what I am saying? Highly stable is hetero. This is unstable because it is prone to reactions. Alright. So here we go. So this tabulates the difference between both. So consists euchromatin. So this is loosely arranged. So consists of thin, extended, lightly stained parts of chromatin. So genetically more active chromatin because it is undergoing transcription and translation. So lies at the center of the nucleus because that is where the action is going on, right? So less histone proteins because it is unwinding. Only if it unwinds from your histone octamer, it can undergo replication, transcription and translation. Okay, so it has less histone proteins and it replicates in the early S phase because it is immediately needed. So consists of thick, this is heterochromatin which is tightly packed. So consists of thick coiled condensed part of chromatin and it is darkly stained. So it's less active or inert. So it is towards the nuclear membrane, meaning towards the periphery. So here you have heterochromatin, here you have euchromatin. So here more histone proteins are there because it is tightly packed. So this replicates in the late S phase. Okay, so this is how your euchromatin is. So this is how your heterochromatin is because your DNA is the information is trying to be copied onto your mRNA. So it unwinds from your histone octamer. Is this clear? Yes? What is the difference between euchromatin and heterochromatin, everyone? No doubt? Shall I move forward? Yes? Okay, so moving forward, talking about chromosome. So the term chromosome was coined by the scientist Waldeyer. Okay, so your chromosome structures are best studied in your metaphase stage because it is highly condensed. So what is metaphase stage? So when your cell undergoes division, so that is known as mitosis or meiosis. So here you have 4 stages: prophase, metaphase, anaphase and telophase, right? So in the metaphase is where your chromosome structures are clearly visible, and that is where you study the structure of your chromosomes. So in a diploid organism, is scientist name important? No. But if the scientist name are mentioned in your NCERT textbook, you might get a statement based question. So which of the following statement is incorrect, which of the following statement is correct? In that cases, the scientist name which are mentioned in your NCERT is important, but the years are not important. Okay, yes. So in a diploid organism, we have 2 pairs of chromosomes, one from father, one from mother. So we are having 23 pairs of chromosomes, right? So in that case, so in metaphase is where your chromosomes will look like this. Your chromosomes will also align together at a particular line, and this line is known as metaphase plate. So this line is known as metaphase plate, alright. So 2n is diploid and n is the haploid number of chromosomes. So here it is diploid. I have 2 copies, right? Okay, so chromosome, so inside your chromosome you have your DNA, so on your DNA you have genes, clear? Yes, so now talking about the structure of chromosome. So every chromosome has a primary constriction. So primary constriction is a small bend. So constriction means a small bend, or it is the centromere. So every chromosome is like this. So your chromosomes bend at this point. So this is your primary constriction, or this is also known as your centromere. So this is the primary constriction of your chromosome, on the sides of which are disc-shaped structures called as kinetochores. I told you what are kinetochores, right? So your centromere holds 2 chromatids of a chromosome. So it holds these 2 and these 2, right? So it holds the chromatids of your chromosome. So this is your centromeric region. So this disc-shaped structure is your kinetochore, so which attaches to your spindle fiber. So these 2 are sister chromatids, and this is one chromosome. So if this is chromosome number 3, so you have 2 copies of chromosome number 3, one from father, one from mother, right? So this is chromosome number 3. If I draw another chromosome and say this is paternally inherited, this is maternally inherited, since both represent chromosome number 3, so they are called as homologous chromosome. So we call it homologous chromosome if it is the same chromosome, but one from father, one from mother. So these 2 arms of a chromosome are known as sister chromatids. Okay? Yes? Done? Okay. So moving further, so kinetochores are the actual site of attachment to chromosomes during your cell division. So apart from your primary constriction, that is your centromere, we also have secondary constriction in your chromosomes. So the chromosomes which have secondary constriction, they are known as nucleolar organizing regions. So like this. So this is the first bend. So this is your centromere. So if they have a second bend like this, so this is known as secondary constriction, or this is known as nucleolar organizing region. So this is known as NOR region. Okay? Yes? Okay. So the chromosome with this secondary constriction are known as SAT regions. They are known as SAT chromosomes or SAT regions. Okay, that is satellite regions. Fine. So at the tip of the chromosome, at the tip of every 46 chromosome, you have repeating structures called as telomeres. you have repeating structures called telomeres. Okay, so this telomere is what prevents the chromosome from further reducing in size. Okay, so telomeres are very, very important. They prevent aging or a reduction in size of your chromosome. So these are your telomeres. So as you age, the amount of telomere decreases. Okay, so that is the reason older people tend to have more diseases. So is this structure of chromosome clear? Yes, so in the last lecture I haven't taught this, so that's why I thought I should complete. So based on the position of chromosomes, we have types of chromosomes. Metacentric is V-shaped, so it is like this. So this is V-shaped, okay. So to draw it more precisely, so this is V-shape, so this is metacentric. So SAT means satellite chromosome. So satellite chromosomes are the ones which have your NOR regions. They are nothing but your repeat regions, okay. The gene for color blindness is located in your X chromosome, okay Vishal. In males, the gene for color blindness is located in your X chromosome because that is sex linked. So submetacentric, so this is L-shaped. So this is L-shaped, so your chromosomes can be of anything like this. Ma'am, so in rheumatoid arthritis, telomere stage will be destroyed, ma'am. Okay, so rheumatoid arthritis, it's an autoimmune disease. So find out what happens in rheumatoid arthritis and what is the role of telomeres. So every time, so telomeres prevent the shortening of your chromosomes. So if your chromosomes become shortened, the gene is deleted. In rheumatoid arthritis, the gene is present, but your immune cells attack your gene product, right? So that is autoimmune disease. So acrocentric is J-shaped, okay. So then the fourth one is telocentric, this is I-shaped. So this is how it is. So metacentric, so exactly in the middle. So submetacentric is L-shaped because one is short, one is long. Then acrocentric, so in acrocentric it is J-shaped. So one is very small, one is very long. So telocentric, you have only one arm, right? So this is your telocentric. Accentric is when you have only one center. So dicentric is when you have two, okay. Accentric is there is no centromere but only two chromatids, and dicentric is when you have two centromeres. So these are the different types of chromosomes. You might get a question from this also. This is also part of your cell, the unit of life. Okay, is this clear? So I guess with this we are done with the chapter. Yes, the last one, karyotyping. So karyotyping is the external morphology of chromosomes. So let me tell you, we have 46 chromosomes are 23 pairs of chromosomes, right? So not all the chromosomes are of the same size and are of the same type. So each has a different shape. It can be metacentric, it can be submetacentric, it can be acrocentric, it can be telocentric. So now how do we know what is the shape of each chromosome? So the study of chromosome morphology is known as karyotyping. So this talks about the number of chromosomes that is present in an individual. So some people have only 45 chromosomes. Some have 47, like your Klein filters. The relative size of the chromosome, position of the centromere, whether it is telocentric, submetacentric, acrocentric, the length of the arm, whether one arm is very big or short, secondary constriction, that is NOR regions, and banding patterns. So this is how you do. The study of external morphology of chromosome is karyotyping. The end result of your chromosome is your ideogram. You get a graph like this. So from here you will know, can you see there is an abnormal chromosome? So this is your Klinefelter syndrome. You should have only one X or one Y or 2X, right? But you have here 3 sex chromosomes. So this is how abnormal chromosomes are detected. So this graph is known as ideogram, and this study is known as karyotyping. Number of autosomes in humans. So we have 46 chromosomes, so out of which 44 are autosomes. So which means you have 23 pairs of autosomes. Okay Vishal? Yes. So this is known as your ideogram. Yes, any more doubts? So with this, I guess I am done with the chapter. Oh, one more slide. So yes, microbodies I have already discussed. I have discussed what is peroxisome, what is spherosome, what is glyoxysome. So they all come under your microbodies. So peroxisome is in plants. Spherosome also is in plants. Glyoxysome also is in plants. So they have certain lipid synthesis, right? And one pair of sex chromosome, not allosome. One pair of sex chromosome. Okay? Yes, so microbodies. So these are the microbodies. Transmosome is in case of birds. Transosome is in case of birds, but this is not discussed for your need, so you can leave it. Okay? Yes? Done? So this chapter is done. Any doubts? So should I discuss anything once again? So please take a minute to understand whatever we have been discussing. Yes? Any doubts? Nope. Okay, so shall we go into questions? So please evaluate yourself. At the end of all the 15 questions, you will give me how many right answers, how many wrong answers you opted. Okay? Yes. So an organelle devoid of membrane covering is vacuole, ribosome, peroxisome, lysosome. Quick. Quick. Okay, Keshav, yes, perfectly right answer. Others? Others? So Keshav has given us the first right answer. Yes, you're right, Keshav. Okay, others, what is the answer? Yes, perfect. Ribosomes are non-membrane bound organelles which are present both in your prokaryotes as well as in your eukaryotes. So vacuoles have a membrane that is known as stonoplast. So peroxisomes are plants. They are also membrane bound structures, but we didn't read about the name of the membrane. So lysosomes are single membrane bound which consist of your hydrolytic enzymes. Right? Okay, so here we move to question number 2. Yes, lysosome is single membrane bound. Yes, you're right. Okay, so difference between prokaryote and eukaryotic cell is in having cell wall, nuclear membrane, ribosome, none of this. So this is a very easy question. Come on. Come on, this is a very easy question. What is the answer? So the difference between the prokaryotic and the eukaryotic cell is perfect. Nuclear membrane, Shimpi, very good. So this is an easy clue. All right. So here I move to question number 3. So the active transport across biomembrane involves production of ATP, requirement of energy, production of toxin, release of energy. Yes, question number 3. 2B, yes, right. So 3, what is the answer? So Prince, hi. So thank you for joining my session. Yes, for active transport across biomembrane, you need requirement of energy. We do not produce energy, so this is wrong. So it is requirement of energy. Yes, very good. Shimpi, release of energy? No, you need to provide ATP. ATP will not be released, okay? Yes, question 4. So function of rough endoplasmic reticulum is autolysis, protein synthesis, lipid synthesis, carbohydrate synthesis. Yes, function of rough endoplasmic reticulum is, come on. So these questions you just need 2 seconds. Yes, 4B, protein synthesis. It is understood because it is rough endoplasmic reticulum, right? Okay, so these are particular chapter tests, so that's where I get the questions from, all right? Okay, so question 5. So centrioles and centrosome occur in the cells of green plants, animals, cyanobacteria and bacteria, both B and C. Smoothest lipid, yes, Keshav, right? So question 5, what is the answer? So question 5, what is the answer? So everyone says B. Yes, so centrioles and centrosomes are found only in your animals. So centrioles are the one, centrosomes consist of centrioles. They help in the formation of spindle and they pull the chromosomes apart. So this happens only in case of animals. So this does not happen in case of plants, neither in case of bacteria. So it is option B. Yes, question 6. Chromosomes having equal or almost equal arms are called. Just now I taught metacentric, acrocentric, polycentric, acentric. Yes. So question 6. So question 6. Come on, quick. Metacentric. Yes, perfect. Very good. So in metacentric, it will be exactly the equal size, right? Yes, perfect. So question 7. Satellite means the terminal part of chromosome beyond secondary construction, terminal part of the chromosome beyond primary construction, terminal part of the chromosome beyond tertiary construction, none of the above. None of the above. So satellite means terminal part of chromosome beyond secondary, beyond primary or beyond tertiary construction. So this is primary construction, this is secondary construction, this is also known as NOR regions, nucleolar organizing region. So this region has a lot of repeats, so that is your SAT or your satellite. So this is beyond your secondary construction. All right. There's nothing called as tertiary construction. Primary construction is mandatory. Some chromosomes might have secondary construction. There is no tertiary construction. All right, Anthony? Yes, yes, Vishal, you're right. So question 8, grana are protein storing plastids, colored plastids, stacks of thylakoid, individual thylakoids present in stoma. Yes, question 8. Yes, question 8. Yes, they are stacks of thylakoids. So this comes in chloroplast. So your chloroplast is double membrane structured. So you have an outer membrane, you have inner membrane. So this space inside the chloroplast is known as stroma. You have disc-shaped structures. This is known as thylakoid. So inside the thylakoid, it is known as lumen. So stacks of thylakoid is grana. So the granas are connected together by stromal lamellae. Okay? Yes, all of you are right. Very good. So question 9. A component of cytoskeleton is microtubule, bone, chitin, cartilage. Very easy questions, right? So please don't miss any question from this chapter. It is pretty easy. A component of cytoskeleton is? Okay, so Prince is your cousin. All right. Okay, so hello. Thank you, Prince. Thank you, Vishal, for letting your cousin know. So thank you, both of you, for joining my lecture. Yes, microtubule and the protein is tubulin. Yes? Clear? Yes, this is option A. Very good. So question 10. Axoneme with 9+2 microtubular arrangement occurs in. An axoneme with 9+2 microtubule arrangement is cilia, flagella, both A and B, or centriole? Yes, Prince, you're absolutely right. So cilia and flagella have 9+2 arrangement, whereas this has 9+0 arrangement, right? So they form the basal body. Yes, very good. So everyone has understood the concept pretty well. So question 11, which of the statement is false? The ribosomes of a polysome translate mRNA into protein. Mitochondria divide by fragmentation. All cells arise from pre-existing cells. The lipid component of the membrane mainly consists of phosphoglycerides. So which one is the wrong statement? Yes, question number 11. Yes, Shimpi, why question number, statement B is wrong? So this is right. So ribosomes help in translation of mRNA into proteins, but mitochondria does not divide by fragmentation. So it's a normal cell. So mitochondria is an organelle, right? So it is formed after your DNA is replicated. So cells arise from pre-existing cells. So the lipid component is phosphoglycerides. Yes, perfectly right. So question 12. Which of the following statements are correct here? Na plus K plus pump is an example of active transport. So in plants, lipid-like steroidal hormones are synthesized in your smooth endoplasmic reticulum. So in plant cells, the vacuole occupies only 10% of the volume. So chlorophyll and leucoplasts are responsible for trapping light energy, responsible for photosynthesis. Yes, Virchow said cells exist from pre-existing cells. So which of the following statement is right? Yes, Prince, you're absolutely right. So you're giving me right answer. Very good. So this is the correct statement. So Na plus K plus pump is an example of active transport. In plant cells, ripples like steroidal hormones are synthesized in smooth endoplasmic reticulum. No, it is in your animal cells. So in plant cells, vacuole occupies almost 90%. So what is responsible for trapping light energy essential for photosynthesis? Not chlorophyll, not your leucoplast, but this happens in your chloroplast. So in your chloroplast, what is present? Your stroma and your thylakoids, right? So your thylakoids are responsible for trapping energy. Okay, so here it is option A. So question number 13, which of the following is wrongly matched? Which of the following is wrongly matched? Chloroplast, chlorophyll, elaioplast, starch, chromoplast, carotenoids, amyloplast, carbohydrates. So which is incorrectly matched? Yes. Which is incorrectly matched here? B. Elaioplast is fats and oils. It is not starch, right? So this is incorrectly matched. The rest are right. No, amyloplast is carbohydrates. That is right. Amyloplast is carbohydrates. Why it is wrong? Prince, we want the wrongly matched statement. It is not D, it is B. B is wrongly matched. Okay, Prince? Yes, oils. So question 14. So which of the following pairs is not correctly matched? So cristae, the tubular structure formed by the folding of inner membrane of mitochondrion. So this is right. Plasmodesmata, the membrane surrounding the vacuole in plants. So plasmodesmata is a cytoplasmic connection, right? So two plant cells or more are connected like this. This is cytoplasmic connection. So this is your plasmodesmata. Right? So grana, membrane bound disc in chloroplast that contain chlorophyll and carotenoids. So middle lamella, layer between adjacent cells in plants derived from cell plate. Yes, so which one is not correctly matched? Option B, right? So plasmodesmata is cytoplasmic connection. It is not tonoplast. So Shimpi, answer is option B, not D. Okay? Yes? So moving to the last question of the evening. A and B, why? So this is right. This is a right question. Why, Vishal, A? It is only B. B is incorrect. Okay, Vishal? Yes? So last question. Which of the following pairs are correctly matched? Microtubules, structural components of cilia. Centrioles store hydrolytic enzyme. Amyloplasts store oil and starch in plants. Yes? Yes, the last question. What is the answer? Tell me. B. A and B are right. So centriole stores hydrolytic. The options are here. So these are the options. So statement B is wrong. Centrioles do not store hydrolytic enzymes. So they are lysosomes which store hydrolytic enzymes. Yes, only option A is right. So C is the right answer. Yes, absolutely right. So we are done with this chapter. So on Wednesday, I'll be starting biomolecules at 6 p.m. So please join me for the same. All right? Yes? So this is my schedule for tomorrow. So tomorrow 4 p.m. we are talking about plant growth and development, and 6:30 we have a mentee. So it is guess the chapter. So it is episode 7. So please join me for the same. Okay? Yes? Fine. So if you genuinely liked my teaching, so please do like, share and subscribe, and let me know if you have any doubts, any suggestions. So you're free to share your suggestions with me. All right. Yes. Thank you so much for making my sessions interesting. So wishing you most and most. So this is me signing off. So have a great evening ahead. Bye.